I'm Liz Maynard, Extension Specialist for Vegetable Farming in the Horticulture Department at Purdue. And I really appreciate everyone being here today, whether you're watching or listening or watching later. I know on a vegetable farm, things are really busy this time of year. So please, if you have questions, put them in the chat box, email us later, contact us later, whatever. My contact today is growing media for vegetable transplants, and in particular media that's acceptable for use in organic production. And there's so many different kinds of media. Sometimes, you know, you wonder, how do you figure out which one is going to be the best one for you? And then the media costs different. So if there's one media that seems better, is it worth paying extra for a more expensive one? We did some work looking at different media in greenhouse trials and planted some out into the field. And that's the main thing I'd like to share with you today, is take you through some of those trials. And I'll start by looking at some of the media we used and using that to kind of talk about what's in a media. Oh dear, let's see, why doesn't this, there we go. So one of the first things to do to get a handle to help you pick a media is just look at what information there is from whoever's supplying the media, the manufacturer. And they'll usually have information about water holding capacity, aeration, drainage, whether there's fertilizers in there. Sometimes they add biological amendments and sometimes other stimulants. So if we look at the media that we used 8 media in our trials. And on here, I've kind of tried to list the ingredients, some comments from the company. And in the ingredient list, the things in blue, peat, that would be something that will hold water. Compost also holds water, but is also a source of nitrogen and other nutrients, so it's in green. Fish meal is in green, a source of nitrogen and other nutrients, and then better drainage. So those color codes will kind of let you know what's in the different media. Another thing to be aware of about media is that getting it wet is really important. Some organic media do not contain wetting agents, and those are particularly important to moisten before you fill the flats. But even if media does contain wetting agents, that's a good idea to do. So we use Johnny's, and the white code up at the top is going to be how I'll refer to them when we get into looking at the experiment, so pay attention to that. We had Morgan Composting Premium Flour 201. Now I think it's just called Flour 201. Again, that's one with a base of peat. It's got some nutrients in it. The text in brown here are items that provide mineral nutrients, and it's got some perlite in it for drainage. And they mention it naturally retains water, so it's going to have a good water holding capacity. We use Penn Valley potting soil. Again, that's got peat and coir, another thing that provides moisture holding. Compost, some other minerals, perlite for drainage, and they mention it's good for, you get faster germination, you won't need additional fertilizer, and they also mention pre-wetting before putting into trays. This ProMix MP Organic had peat and coir, perlite for drainage, vermiculite that also provides drainage and airspace. It's also a source of potassium, but not much else in the way of nutrients. They do add some mycorrhizae, and they specifically mention it's good for seed starting. The Seed Catapult from Ohio Earth Food. This is another peat and compost base. It has both perlite and vermiculite in it. Some other nutrient sources here. And it's one of the ones that had sand in it in addition, provides some additional drainage probably, and they add mycorrhizal fungi. They say it's good for seed starting and will also feed the plants for up to 8 weeks. We had Sunshine number 1 or Sun number 1. It's another peat perlite mix basically. It has a starter nutrient charge, means it's just got a little bit of nutrients in it, but wouldn't be expected to keep the plant up until you would transplant it probably. This one did have an organic wetting agent in it. Vermont Compost Fort Light, another one with peat and coir, a number of ingredients that supply nutrients. They mention it's good for cell tray seedlings, and they do mention wet before planting, and they say it drains a little better than another media they make called Fort V. We did have Fort V in what I'll talk about today, and that is very similar, but you'll see it doesn't have the perlite and vermiculite. And they mentioned that it has good water retention. So those are the 8 media that we trialed, and I'd like to go on to kind of run you through a trial that we did. We did trial them with a number of different crops, but today I wanted to show you the lettuce trial because it's fall, and we actually started these lettuce in late August in 2018. This is what the trial looked like as we were starting out. Each media was in a half of a 128 cell flat, so it had 64 cells in there.
Liz? Hello?
Yeah.
So at this point,
yes, Dan.
we were talking yesterday, and you had talked about wetting up the media, but if you look at these trays, it looks like they haven't been wet up.
Yeah, that's a good point. In this study, what we thought, should we wet the media first or should we not? And we thought, well, rather than wetting the media first, we're going to put it in the trays, and then we're going to after we seed, we'll water all the trays until the water runs through. So we tried to get them all wet. And when we repeated the study later times, we did wet the media in advance. But in this first run of it, I guess we're worried about whether we were getting the same amount of media, water in all the media, so we didn't do it. But some media, it works better definitely to wet before. Some of the media that has wetting agents in it, you can do just fine with watering after it's in the trays. So that's a good point, though. We started taking data as soon as they came up, and these are the 8 media just as they're coming up. And for lettuce, it was interesting. The 2 media with stars there, the Sun one and the Vermont Compost Fort Light, they actually came up on average a little bit quicker than some of the other media, and then the PAN and the PMPO were the slowest. With tomatoes and the other crops, we saw a little bit different pattern there, but there wasn't a huge difference in emergence time for lettuce. And then we, by 10 days after seeding, already you can see differences in growth there. If we just go through and look at the particular media here, if you remember what was in some of those different media, you might, you might say, oh, I'm not surprised that PMPO was kind of small. That was the one that didn't have very many nutrients.
One thing, Liz?
Yeah.
So one thing we talked about yesterday is, at this point, I was kind of wondering, is bigger necessarily better at this point?
That's such a good question, because sometimes when you're growing a crop, a seedling crop, you just, you don't want it to grow too fast. The seedlings will get too big and maybe too lush for transplanting. But for lettuce, and we think of that maybe also with like cucurbic crops or tomatoes, but for something like lettuce, where I just want that crop to get up and be able to harvest the leaves, I don't know if I've seen studies about it, but I think it's fine for it to be growing quickly at this point. And then 18 days after seeding, they look like that. And another, there's the individual trays. And then by 23 days after seeding, this is about when we went to transplant them. There's different ways we could have done this. We could have just grown them all, you know, at a certain size and transplanted each at a certain size, but we decided to grow them all for the same amount of time and then transplant them. So this is what they looked at like. Yeah, and then we put them out in the field on September 13th there, and got them in the ground. And a few days later came back and noticed that some deer had come in and had salad dinner from our transplanted lettuce. So we still had transplants left over, so we put those out and we put them under a row cover to protect them from deer, and that worked pretty well. And so the plants were a little bit bigger by the time we put them out. Those under the row cover, they established well. Even the tiny little plants, you can see some here that are quite small, those established quite well. And we did a first harvest of them on October 11th, and then came back and did a second harvest on the 18th. And on the 18th, we harvested both from under the row cover and plants that had not been chewed extremely by deer outside the row cover. We harvested those as well. You can see in our field, we have a wonderful stand of annuals coming up, and we also have a lot of volunteer hairy vetch because we use that as a cover crop. So here's our lettuce where we're harvesting that was under the row cover. And then these are, I want to show pictures of what the different treatments looked like. So we had harvested every other plant before, and we were coming back and harvesting these. These aren't necessarily full size for a romaine lettuce, but they are, you know, of a sellable size if you wanted to sell them. You see what the size of the plants look like. So from this, I was able to calculate yield based on a weight per head. And in the next slide, I'll show you the results of the yield, for the different treatments. So this is the lettuce weight in pounds per plant or pounds per head from that October 18th harvest. You'll see all of the ones with compost, media that had compost, did better than the two media without compost, the PMPO and the Sun one. And of those media that had compost, the Vermont Compost Fort V did better than the Johnny's 512 and the Flower 201, Morgan Composting 201, and the other ones were intermediate. So that gives us an idea of, well, which one might perform better just in terms of yield. But of course, not only yield, it's also the, how much might you be selling that for. So in this next graph, you'll see the bars kind of have the same relations to each other, but on the y-axis here, it's the lettuce value per 60 quart of bag. So assuming you would get 2,048 plants out of a 60 quart bag of media, and assuming you're selling lettuce at $2 a pound, that's the value that you would get. The other thing you see on here are these wide parts of the bars. That is how much the bag of the media cost. So you can see differences in the cost of the media from the two Vermont composts are higher cost. Sun One was the least expensive. PMPO was also pretty inexpensive. And so with those two numbers, the value per quart of 60 quart bag and the cost per 60 quart bag, you can figure out how much value you're getting for every dollar you invest in that media. And that's what this graph shows. The bars represent the dollar in lettuce sales per dollar you spend on the media. The little lines here are the cost of the growing media. So here you see the media kind of fall out differently. The one that's highest is the seed catapult. The media didn't cost too much and it had a pretty decent yield. So the lettuce value per media, per dollar spent on media is higher. The Vermont Compost Fort V, because its cost was so high, you get less dollar value for every dollar you invest in it. So this is another way to think about and evaluate. I have to make the point that your media cost for these particular media will make a huge difference in this graph. These media, you can buy them in bulk totes. You know, I did not include shipping costs in this, in the cost of the media. And of course, the price of the lettuce that you're selling and the how many lettuce plants you get out of a certain volume of media, all of that will influence how this graph sorts out. I just wanted to show that to give a different perspective than just looking at the yield. I wanted to mention just a little bit about adding nutrients, because I think the media that did not do so well were also the media that did not have as many nutrients in them to start. And you can add nutrients by incorporating organic fertilizers into the media. That needs to be done with real caution so you don't get in too much. You can top dress things that are high in nutrients, just sprinkle it on top. And you can also add liquid fertilizers into irrigation water. And I think, yeah, so we did another trial with tomatoes. We add nutrients to some of the media, and this little video I took of that trial. And you'll see here the abbreviation for the media and then fertilizer added. And the 524 is a fertilizer that we just top dressed. And then there's another one that we put in irrigation water. So let's just look at this, see if this will play for you. Here's the sun one with some fertilizer. Vermont Compost Fort Light with fertilizer. Here's M201, no fertilizer. There's the SCOE, no fertilizer. Vermont Compost Fort Light, no fertilizer. There's that Pro Mix, no fertilizer. Vermont Compost Fort Light with liquid fertilizer. M201 with some fertilizer. Pro Mix with liquid fertilizer, and then Pro Mix with the top dress fertilizer. So the fertilizer did definitely help. And oops, I didn't mean to play that again. Stop. And that's something I would recommend if there's a media that's low in fertility, or if it looks like your fertilizer is running out. In terms of using liquid fertilizers, it's helpful to know the NP and K that's in your fertilizer, and these are some common ones. There's certainly other ones you could use, but so Neptune's Harvest, for instance, is 2% nitrogen, 4% phosphorus, 2% potassium. And just realize that something like this fish fertilizer here doesn't have any potassium, phosphorus or potassium, and if your media does not have that to begin with, and you're only using that, you might not get results that you want. But if you start with a low nutrient media, like the ones we had were the Pro Mix and the Sunshine #1, I'd recommend applying a liquid fertilizer as soon as you see those first true leaves develop. In fact, these tomatoes were already a little bit behind. If you have a media with more nutrients in it, I would apply based on the crop appearance, and that's something you'll have to learn from experience. But this shows some Vermont Compost Fort Light without any additional fertilizer added yet, and the leaves are a little paler green, and you can see a little bit of purpling compared to the media on the right where some fertilizer had been mixed in before. So if you have a higher nutrient media, I'd wait a little bit, and based on experience, apply additional nutrients later. I just discovered this information resource on organic potting mixes written by Dr. Michelle Wander at University of Illinois, a nice summary at eOrganic. It's at node 3442, it's a resource you can look at. And then just like to wrap up by saying that picking a media that is suitable for your operation and managing it well, adding nutrients if needed, is really a way to get off to a good start. So know your growing media, choose one that'll work for you, look at your plants. If there's an issue, address it quickly, add nutrients if needed. And I think Dan's going to talk about looking at your plants. And I think that's the last, yeah, that's the last slide that I have. So I'll.
Okay, so Liz had a lot of nice, wonderful slides, and I'm afraid that a lot of mine are not going to look so pretty. So what I want to talk about is, let's say you're growing things and you have a problem. Certainly, I won't be able to go over every possible thing that can go wrong and identify here, but just kind of help you to kind of troubleshoot what might go wrong. And the first slide, Liz and I kind of went over these together yesterday, and this kind of overlaps pretty much with hers, and I don't know if Liz wants to comment, but so what Liz talked about was certainly if you buy different mixes, she showed clearly that there's differences within the different trade names, but this actually is differences within one bag. So a couple points here, just that from a disease standpoint, if you look at this, and this is one of the important things, is distinguish what is a disease and what isn't a disease. And even though the one on the left is stunted, it's not diseased. It's a media problem. And if you look overall, here's this is from a commercial outfit, and you see there's great differences in the seedlings, in the size, which is primarily, and you can tell there's by tray pretty much, it's different bags and mixes, whether that was different quality control or whether some of them got wet ahead of time. But then also sometimes this is growing on gravel, and you can kind of see that you can kind of see that there's differences in elevation, and that'll affect how seedlings grow as well. But really, most of my talk, what I wanted to devote to kind of troubleshooting again. So here you have a tray, it's turned yellow, you're upset about it, you want to determine what it is. And one of the things you want to determine is it a disease or not? Because you know, if it is a disease, it may spread within the greenhouse, and if you put it in the field, it may spread. If it's not a disease, it may still be important, but it requires different management. And then you may want to know whether it is a problem of a foliar disease in the top of the plant or something down under. So you look at this tray, you see yellowing, and we're going to kind of close in and look at that. And if you look at a close-up of one of those seedlings, then the first thing we can see is there is no obvious lesions, no symptom or sign of disease there. It's just a kind of chlorotic or yellow and wilted. But also, and Liz and I were talking about media and drainage a little bit yesterday. Notice that the media looks very wet here. And in fact, it looks green, which to me indicates probably it's either been overwatered or there's or it just doesn't drain very well. And in fact, in one of the screens, there is one of those cells, there's a little weed growing. So at this point, I would say it doesn't look like a disease, but the other thing you want to do is to lift up that seedling and look underneath to see what it looks like. And one of the things you might see is that there's a root problem. This may be a disease, or it may be, again, a media problem that's causing the roots not to grow, such as too much water. But if you look on the left, clearly this root is not growing well, and so the root problem would probably explain what's happening on top, whereas the one on the right there is growing very well, and the roots look nice and white and have filled the container. This is, if you talk about greenhouse problems, this is probably the most common damping off, in this case of cantaloupe, but it can happen of pretty much anything that you want to grow in a tray in a greenhouse like that. But there's any one of a number of fungi which might attack the seedling at the soil level, which has happened here. You see the lesion. So you may buy a bag like Liz talked about, how much trouble you go into to purchasing a bag, and how you might bring it home. And then if you dumped it all in a greenhouse floor that's not sterile, or used a shovel on it that's not sterile, then you may have contaminated it with one of these fungi, which may go in and attack the plant such as you see here. But also the soil, this mix here looks pretty wet. So wet, cool conditions also favor damping off. All right, so I just wanted to point out here that in this case, you can actually see lesions on the plant, and note that and so this is much more likely to be an infectious disease. And again, the soil looks very wet.
So I wonder if, Dan, this is Liz, but I'm wondering if, since we didn't get to see all the pictures, whether it would make sense to just go back to the beginning and go through them again.
I don't know. This is the first one of the earlier slides here. Let's see if I can do this. And this is, let's see. So this is the case. In this case, you don't see any, I don't see any lesions on here. So I would assume that this, at least at the top, looking at the top, there doesn't look like a disease. But if you look at the bottom, then you can see that, well, maybe the lesions on the top are caused by a root problem. And you see on the left that seedling does not look, the roots are not healthy, whereas the one on the right does look healthy. So just a reminder you might want to lift up some of those seedlings and look at the roots. And this is damping off, classic damping off symptom at the stem here. And this is cantaloupe, but any number of crops and even bedding plants can have damping off. Now this is the second one. This is, again, you look at its yellowing, but as we close in here, you can see again the lesion on the seed leaf there. And to me, this, even if you didn't know what that was, it would indicate that perhaps maybe there's an infectious disease going on. And again, that is what's going on. But as Liz and I talked about yesterday, there may be more than one thing going on because the media looks very wet. And there's also some yellowing that probably is unrelated to the disease. But this is gummy stem blight on cantaloupe. In this slide, all I wanted to do is just kind of make a note the distribution here, that a lot of times diseases will at least initially be clustered. So that's kind of maybe a hint that there's an infectious disease going on and not some nutrient problem like maybe that Liz was talking about. And again, kind of following that same troubleshooting scheme, if you look, as I look at the very top of this seedling, the shoot, and you see the dieback here, first thing I would suspect is the root. But as you look at the roots look healthy here. So what I would do then is to look at the inside of the stem, and you see the top of the top seedling is healthy. The bottom has a vascular problem. So there's a number of ways to troubleshoot and a number of things that can go wrong there. This is a foliar disease. This is anthracnose, a watermelon. And here what I'd like to point out is looking just at the different sizes of the lesions and the different shapes. And this, if you think about an infectious disease, this is the kind of thing that you would expect to see different sizes and shapes of the lesions based on how long those lesions have been growing. In contrast, this is gummy stem blight of watermelon. Again, you have several different sizes and shapes, but notice that the lesions don't all occur along the edge of the leaf. Something that occurs along the edge of the leaf is probably not a disease. Here's just looking at one individual lesion. This is angular leaf spot on watermelon. Something that has a halo like that, a yellow halo, probably it might make it more likely that it's a disease. And look at the sharp corners of the lesion, and then the lesion itself varies in coloration within the lesion. In contrast, this is a non-infectious problem, and if you look at how the veins are standing out and the rest of the leaf is affected, this is often a symptom of a non-infectious problem. Again, it may be important, but it's kind of helps you troubleshoot the problem, and you know it's probably not going to spread from plant to plant if it's non-infectious. Just a similar slide picture showing something that's between the veins probably indicates that the problem is non-infectious. And in this slide, and I know this isn't seedlings, too often I don't take photos of lesions that are non-infectious, I guess. But if you look at the top photo, this is classic infectious disease lesions where there's a yellow halo and there's rings or structures within the lesion. In contrast that with a larger photo where it's a herbicide problem, and what you see is just no halos. The lesions are just white, and there's nothing going on in the lesion. So that's typical of a non-infectious lesion, one that doesn't have any growth appear to have any growth in the lesion itself. This one's a media problem. This is actually something was added to the media, and it was poorly calculated, and too much was added. You can see that one of the seedlings has escaped here, but a problem where almost every single plant is affected probably is not an infectious problem. It's probably a media problem. I don't have a photo of the roots, but the roots look okay, but it's probably not getting the necessary nutrients. Here's a close-up, extreme, probably some kind of nutrient. Maybe everything is missing here.
Yeah. One thing I was going to say about that one, Dan, I remember is I had mentioned, well, actually I didn't mention it because I didn't have time, but when media pH is way too high, sometimes you'll see like bleaching or yellowing in the youngest leaves because of micronutrient deficiencies. I don't know that that's what that is in this case, but if you have way too high media pH, sometimes you can see that.
Well, it may be that this miscalculation caused exactly that. But just looking at it, I guess this is a non-infectious problem. And of course, it is. So I showed damping off. Maybe I should have had this photo closer to the other photo where you had a lesion right next to the soil level, and that's classic damping off. But notice that this lesion is kind of starts at the seed leaf and works its way down. So this is not classically how damping off works. In fact, this is gummy stem blight, and you can see in this case it's kind of woody looking. You probably wouldn't plant this, but it's important also to know that this could spread wildly in the greenhouse or in the field. So you would want to avoid both the seedlings that look diseased and probably trays and nearby trays that look this way. And if you kind of go back in time, this same lesion will look kind of water soaked. And if you were to call myself up or somebody and say, well, it's got water soaking on the stem, it sounds kind of like damping off, but again, notice that this is not at ground level. It starts kind of closer to the seed leaf. And again, this is an infectious problem. This is obviously not a transplant tray, but any kind of situation where you see a seedling that's completely yellow next to seedlings that are perfectly green, that indicates probably it's not an infectious, not a disease problem, but more likely perhaps a genetic problem. Liz and I were talking about what this is called, but this is some kind of genetic mutation in the seedling itself. It won't spread. It's not a concern if there's not a lot of them like that. But if you saw that, it might kind of give you some pause as to what's going on in the seedling. In this one, this is a black root rot of watermelon. What I wanted you to focus on here is that there's a dieback. The plants are dying back, kind of turn yellow, and the leaves have died back. What when I look at this, I don't see much in the way of specific lesions, and the first thing I want to look at is the root.
Yeah, you know, Dan, when I look at that picture, I think, boy, that looks like, you know, sometimes I'll have a tray that's just been cucurbits that's been in the cell a little bit too long, and the cotyledons are dying off and stuff because they're running out of nutrients. It almost looks just like that.
You know, and I don't think, just looking at this, I don't think you would be able to tell the difference.
Yeah.
And so what I would say is, my first reaction then would be to pull those up, and I guess I don't have when I first pulled it up, but after I washed away the media, this is what it looked like. And I think you might not know exactly what this is, but looking at it, you can see the root systems are poor, to say the least. But also you see the lower stem, or hypocotyl, has this lesion on it, and clearly something's gone terribly wrong here. Probably the media became contaminated at some point. And that's all I had. There's my contact info, if you want to contact me at any point.
So my name is Kyle Richardville, and as Petrus said, I'm a graduate student. I'm a master's student in Lori Hoagland's lab. And so my research is concerning the mediation of biotic and abiotic stress by trichoderma inoculant in leaf composted soil. And my research is at the Purdue Student Farm, where this Small Farms Conference would have been held. It was held last year, and it turned out great for us, but unfortunately, you know, it's not in the cards this year. Okay. So Lori Hoagland works a lot with urban agriculture, and my research is especially important for urban agriculture as well. And this is just a growing topic for global food security in general. The UN says that over 2/3 of the world's population will live in urban areas by 2050. So there are a lot of challenges associated with getting fresh, healthy produce in those areas. And we see a correlation in urban population growth and urban agricultural growth, especially in the past 30 years. There are so many benefits to these urban agriculture endeavors, but also a lot of potential challenges, as I'm sure a lot of you know. Some of them include compacted soils, low soil fertility, and the potential of contaminated soils. So my research specifically has to do with growing tomatoes. We have two varieties. At the top, we have Corberino, more cherry tomatoes, and the bottom, Wisconsin 55, more plump, round, bigger tomatoes. And so in the middle is a picture of myself holding the leaf compost that we used. We got it from Dan Perkins, a farmer in DeMont, Indiana. So he gets the leaves from the city of DeMont. He said he gets around 80 to 90 loads. He puts the fallen leaves in windrows. I think he said he turns them once or twice a year. And he said it's about 10% grass clippings and other organic material like that, and the rest is dead leaves. And he does that for 3 years, and then the product is this beautiful black compost that you see. And on the right is a microscopic view of the fungal inoculant that we use. It's called Trichoderma harzianum, I believe it's pronounced. Yeah. So why this Trichoderma harzianum? As I'm sure everybody knows, the soil is a living ecosystem. It's often called the microbiome of the soil. It's crucial to plant health and development, and there's so many living organisms. In fact, I think it's said that there are more living organisms in a teaspoon of soil than there are humans on the planet. So it's very important to plant health. So research has shown that this Trichoderma species attacks, parasitizes, and gains nutrients from other soil microbes. And it induces a plant's defenses against harmful pathogens that cause disease. And it likes to grow. It thrives where roots are healthy and numerous. And So not surprisingly, it's developed mechanisms to attack these other fungi and enhance plant and root growth. The picture at the bottom is from Cornell University. So if we look at that, that's a corn plant. On the right is with trichoderma, and on the left is without. So we can see how the trichoderma affected root growth and abundance. And on the right is soybean plants, and you see the same effect. At Purdue, the benefits of this are often in controlled greenhouse settings. So when we move it to the field, the results are variable. And that's where the leaf compost comes in, because we hypothesize that amending the soil with this leaf compost has the potential for increasing the trichoderma abundance and efficacy in the field. A lot of this reasoning is because a leaf compost has a high carbon to nitrogen ratio, and fungal species prefer high carbon to nitrogen environments. Leaves are the most available source of organic compostable material in urban settings. So it's very important, especially for urban agriculture again. It increases the soil's water holding capacity. Some studies show up to 500% of its own weight, which is amazing. And it improves the soil tilth, which is just a scientific term for the soil's suitability for planting or growing the crop. If we look at the picture on the bottom right, on the right you see a soil that's crusted over on the top, which has low soil tilth and a lot of runoff, whereas on the left there's not that crust, and the water can infiltrate and move down where the plant can use it. And so the leaf compost has been found to not only help the water move down, but also hold that water where the plant can use it. And lastly, this leaf compost adds a lot of beneficial organic matter to the soil. A study in Michigan found that there's around a 12% crop yield increase in their study for every 1% increase in soil organic matter. So it has a lot of implications for harvest. And finally, why the two varieties? In our greenhouse studies, we found that Corborina was responsive to the trichoderma inoculation, and the Wisconsin 55 variety was not. So we want to see if the same results are observed in the field. So looking at my experimental design, on the left is a schematic. It's an aerial view of the plots, which you can see pictured on the right. So the white squares denote control or no compost amended soils, and the yellow is compost amended soils. The T rows are trichoderma and the W are non-trichoderma, and G1 and G2 just denote the two different varieties. And so each square has 24 plants times 8 boxes, we have 192 plants. So what did we find? After doing some soil testing, we found that the leaf compost did, in fact, increase our soil organic matter. On average, the composted soils were around 7% organic matter, and the non-composted soils were just shy of 4%. And so if we think about the 12% increase per 1% increase in soil organic matter, I'm sure that tapers off as it goes, but this has the potential to increase yields, which we observed and we'll show on a later slide. When we think about abiotic stress, we were really looking at how the heat and the high wind affected transplant survival from the greenhouse to the field. I don't know about your neck of the woods, but here in West Lafayette, we had very high winds this spring. It happened conveniently right after we planted in the field. So we saw a few plant deaths. But the interesting thing was the plants inoculated with trichoderma survived a much better percentage than the non-inoculated plants. We didn't see effect in compost versus non-compost, and we didn't see any differences between varieties. And And so once they were planted in the field, we wanted to see how well they grew. And so I've been tasked with taking weekly photos of every plant throughout the growing season. And so this is just a snapshot of 2 weeks after planting in the field. So on the left, we see 3 plants in the control group, or no trichoderma inoculation and no leaf compost. As you can see, they're pretty long and spindly and pretty weak looking actually. And on the right, we have plants in the trichoderma inoculation and leaf compost group, so they got both of the treatments. You know, I tried to find an average of the control plants and an average of the treatment plants. This isn't just like finding the worst versus the best. I have hundreds of, of plants that show these results if anybody's interested. And so if we look about 7 weeks later, these are the same 3 plants. Again, with the control plants, we see they're long and kind of spindly, and on the right, basically they grew like wild. If you look at the strings, they're bowing out, and the stake is actually bending inward because there's just so much growth. It's quite amazing to see. And so when we look at the nitrogen fertility of the different treatments, so I used a SPAD 502 handheld meter that tests the chlorophyll content. This is a quick and efficient way to estimate nitrogen status. And nitrogen, as everyone knows, is a very important nutrient, and it's a component of chlorophyll, which is the compound that uses the sun's energy to produce sugars. And so when we look at the chart, we see a noticeable difference between the composted and the non-composted treatments, but very little difference in the trichoderma treatments. And then when we look at biotic stress, which is more living organisms causing stress, we tested this by looking at the percentage of foliar disease incidence, and these values are from July 31st of this year, which was our most current reading for this year. So we see foliar disease pressure higher in the Corborino plants than the Wisconsin 55 on average. We see a lot lower effect in the composted soils. So we see less disease pressure in plants grown in the compost amended soils. And we even see a trend towards lower disease pressure in Corborino grown in compost treated with the trichoderma. But we don't see it in the Wisconsin 55, which is in accordance with our greenhouse experiments. And finally, when we look at the yields of last year, we see significant increases in the yields of plants grown in the compost versus plants not grown in the compost amended soils, especially with the Corborino plants. As you can see on the right of the graph, it was very stark and evident to see. And in Wisconsin 55s, we even saw a greater yield in plots with the trichoderma in the compost amended plots, which was really, really interesting to see. So some conclusions that we've observed from the experiment: the trichoderma strengthened transplants, helped them tolerate the abiotic stress of heat and high wind. The leaf compost greatly improved soil quality. It helped increase yield and seems to improve plant fertility. And the compost helped reduce foliar disease spread, especially when paired with the inoculant. So why should a small farm care? So trichoderma and many other biological or beneficial microbes or biological control agents are going to be increasingly important in the future of agriculture and urban agriculture and organic farming. Hundreds of millions and even probably billions of dollars are being poured into research in this area. So these inoculants increase plant resistance to disease, like we said, and improve root development and plant growth. And the leaf compost reduces drought damage, increases plant nutrient holding capabilities and can dramatically increase soil organic matter. So the bottom line is it can help out your bottom line and reduce input costs, reduce negative environmental impacts, and increase output productivity for you and your farm. My research is supported by the TOMI project, which is the Tomato Organic Management and Improvement Project. Lori Hoagland works very closely with this organization. And if you're interested or want to find out more information, the website is eorganic.info/TOMI. Thank you very much.